Fine day to you folks. Welcome to another ALS Expert Series talk. And today, all I can say is you're going to meet a new friend. There's somebody in the box next to you who's probably looking for an extra smile. So if any of you have them to give out today, please do. I know my friend Dorothea, a couple weeks ago, she said, McFinn, this is my happy hour. And I thought, oh my gosh, for you too, because it's my happy hour. I live deep in the woods and don't see that many people. And when I have all of you show up, it's like, hey, we got a family. Might be really extended, but it's a family. So once again, everybody, welcome to this evening. And with that, I give it to you, Indu.
Thank you, McFinn. And yes, I am in Southern California, so it does look warm here. And so I'm really excited to invite, Alon, who is the CEO of NeuroSense. And more than anything, I would call him a friend because, Alon is joined, NeuroSense has joined to support everything ALS Consortia. And this is to help with this groundbreaking open innovation platform that we are building, and can be more thrilled to have a partner. And you will witness today through the presentation of NeuroSense and what the team and the CEO, with the leadership of Alon as the CEO, has been able to build. So with that, I would love to kick off today's event with James doing the introduction. And after that, we're gonna have a presentation and then the Q&A, moderated Q&A, and then an open forum. So welcome, and we're really excited for today's evening. James, take it away.
Hi, everyone. Welcome back. And for those of you who are new, welcome. Today's guest, I'm going to read a little bit of bio and introduce him. Prior to the establishment of NeuroSense, Alon founded MediCan Consulting, a successful consultancy firm which worked and collaborated with many leading pharmaceutical R&D companies, including Mediwound, Chiasma, Teva, Solgel, FutuRx, NeuroDerm, and others. The firm's diligent employees were pioneers who paved the way to more efficient and accurate work in the industry, mainly in small biotech companies, while coordinating innovative drug development programs, managing operational activities, and business development. Alon holds an MBA and BSc in industrial engineering, both from Ben-Gurion University. And without any further ado, I would love to welcome today's guest, Alon Ben-Noon.
Thank you very much, James. And thank you, Indu. It's an honor for me and for us at NeuroSense to be friends with an organization that takes care of patients who suffer from ALS and does miracles in the field, advancing the field. So it's great to be your friend. It's an honor to be a friend. And I'm also excited to be here with you, speaking to all of you guys. I can't say that I'm used to it, but of course I experienced several occasions where I spoke with in such occasions or other occasions that we put out. Each time I'm excited about it, seeing the people that we are working for. We are here working for you guys. That's what we are doing, and you are the energy that drives us forward with what we are doing. So I'll put out the presentation. Let's see if I can share screen. I can. And
as you're bringing up, Alon, I just wanted to mention that your commitment shines through. You're here in the middle of the night in Israel. So thank you very much for joining and doing this in the middle of the night for you.
Thank you, Indu. Of course, not an issue at all. It's just I want to apologize for my broken English, which is not perfect even during the day, and definitely not at this time of the day. So apologies for if my accent sounds bad or my English is not perfect, and it's not. So I'll start with the presentation, and I'll be honored, of course, to answer questions afterwards. But if you feel like you need to ask a question while I'm speaking, no question about it, just do it, ask, and I'll do my best to answer. Okay, so NeuroSense Therapeutics is also a Nasdaq company now since December 2021. This is why we have this fancy disclaimer. I'm not expected to share any information, new information that wasn't shared before, but still we have this disclaimer. I think that the most important part at NeuroSense is how everything began with a chance meeting between myself and a very special person named Shai Rishoni, whom you see here in the photo. And this is a phrase from Shai: "I may be paralyzed, but I can move mountains." And that was true. When I met with Shai for the first time, I was amazed by what he was doing, the capacity of what he was doing to advance matters in the field, either assistive technologies or therapies for people like him. And he knew that at a certain point that he would not be able to enjoy from any therapy since he was already completely paralyzed. And also by the time that I met with him in early January 2016, and then I met him with many more times. One of the time was, I don't know why we have this here, this line, this red line, but one of the times was with my, several times with my daughter. She was baby at that time. He was very excited and touched by that. Basically, as I mentioned, I was amazed by his capacity, what he was doing in the field, and I was a little bit even, how should I put it? Maybe it's not the perfect word, but a little bit embarrassed by the fact that I was doing, relatively a lot, but definitely not enough, not for a certain specific cause. And he never told me, Alon, do something about ALS and go find a cure or go find a therapy. But in many ways, he did so by his personality. And so we started thinking about me and colleagues of mine, great scientists. I gathered several of them, talking with them about Shai, about ALS, and we're trying to think, what can we make different in this field? How can we tackle this huge problem, huge puzzle and complexity of ALS? And of course, it started with the mechanism of action or with the pathological pathways of this disease. It was clear to us at that time that it's not just one. We can't tackle just one mechanism, but we need to find the most relevant ones and to tackle several of them at once. And maybe by that, we can create a clinical benefit for the patients. So we identified several mechanisms. I'll mention them. And then we identified also the molecules that might reach to these targets and create the required change. I'll just mention that unfortunately, Shai passed away in May 2018. But before he did, we already started enrolling and moving forward with the preclinical studies. And we had outstanding results. The preliminary results coming from the preclinical studies that we conducted were by all means outstanding. I came to Shai's home in early 2018 and told him about it. He was very happy to hear. And then I told Shai that since we understand that we have a therapy that we can and we must move forward now, we are going to name it, and we'll name it after Shai, as a tribute to Shai. His last name, Rishoni, translates to prime in English. So we chose prime, prime C, prime combination, and also since C represents both compounds that we're using as a part of the unique formulation that we developed, ciprofloxacin and celecoxib, two known molecules that we combine together. Okay, so moving forward, so we started with the developing a formulation, a novel formulation of two FDA-approved drugs to target multiple pathways. We conducted preclinical studies both on the SOD1 mutant and the TDP43 in 2 different labs, one in Israel, the other one, the TDP43 at McGill University in Canada, with outstanding results in both models. We're very excited about it. Also, not only from a behavioral perspective, but also from a morphological perspective, since we saw changes in the cellular level. So that was great, and we pushed this program forward very fast towards the clinic. We started a proof of concept study, which I'll mention soon, a clinical proof of concept trial, phase 2A, that concluded successfully, and now we entered into a phase 2B clinical trial, Paradigm, that's the name of the study. And this is a 505(b)(2) pathway, which means fast or expedited to the market, expedited program to the market, because we have lots of safety data with regards to PRIME-C. Patents were granted, and we started developing additional programs for Parkinson's and Alzheimer's based on the knowledge that we gained from ALS. But of course, our main focus and the most important indication for us, for me personally, is ALS. So this is PRIME-C, a nice oval blue tablet, a sophisticated extended release formulation that combines ciprofloxacin and celecoxib in unique doses, and it is supposed to maximize the synergism that we found to have between these 2 compounds. The main targets are regulating microRNAs, affecting intracellular recycling, and reducing neuroinflammation, hallmarks of ALS. And this is something trying to describe the mechanism of action with PRIME-C, and what we are affecting, what we found that we are affecting, reducing TDP43, which is a toxic protein, as I'm sure some of you know and are aware of, regulating microRNAs and reducing neuroinflammation, which is in the surrounding of the motor neuron cells. So part of this mechanism is required, or actually relates to the motor neuron itself, and others to the surrounding of the motor neuron, to preserve it as much as we can and maintain the functionality of the motor neuron, which of course activates the muscles. So just a brief overview of the preclinical studies that we conducted. We ran studies first with celecoxib, different doses. We didn't achieve much with celecoxib alone. We didn't expect to achieve much with celecoxib. We know also, we are familiar with the study, with the clinical trial that was conducted at phase 3 20 years ago with celecoxib in ALS population, and didn't gain anything, didn't show an effect. So we see it very clearly here in the zebrafish model. And with ciprofloxacin, the results were similar to riluzole, which all of you, of course, are familiar with. But the combination of cipro and celecoxib together, PRIME-C, was shown to be to work synergistically and more than twice better than riluzole. What does it mean clinically? We don't know. This is what we are looking now. What does it mean being better, more than achieving results more than twice as good as riluzole in a zebrafish model? You can see a swimming pattern here of the healthy fish as opposed to the fish with ALS, but once treated with PRIME-C, you can see that it gains, that the swimming pattern looks much more like the healthy fish. And as I mentioned, the cellular level, you can see a motor neuron of a healthy fish as opposed to the sick fish. And once treated with PRIME-C, the motor neurons looked much more similar to the healthy ones in all parameters that we measured, statistically significant. So basically they are the same, this and that. So we were super encouraged by these results. We moved forward as fast as we could towards the clinical stage. We started a phase 2A clinical trial, one arm with 15 people with ALS. We dosed the participants for a period of 12 months, and we were looking at safety, mainly safety, because that was a proof of concept, just first verify that the drug is safe. Efficacy, of course, the ALS-FRS, vital capacity, and the biomarkers. We did a study in Israel at the Tel Aviv Sourasky Medical Center. And these are the results. I have to mention that this was done with an intermediate formulation and dose. So it's not the final formulation that we're using now. It's not the blue tablet that I showed you just before. And we still gained a very meaningful result with 30% difference between people who were on patients who were on prime C versus matched patients that were identified in the product according to 9 different categories, parameters. So we found patients who matched our patients and participated in clinical studies in recent years, who were on placebo arms, of course, in these studies. And we compared these patients to ours, and 30% difference in the vital capacity and 18% difference in the ALS-FRS, which in my perspective, in my view, way not enough. We must achieve better results, and we think that we can achieve a better result because now we improved both formulation and optimized the dose for the next study. So we have high expectations. I personally have high expectations and high hopes about this study. Now we'll need to see that if we are correct and we can get better results here. It's still important to note that all the patients that completed the study after 12 months requested to continue on prime C, and at least in Israel, what we are familiar with is that most of the patients in other studies didn't really care to continue with the experimental medication. And here, this was very encouraging. And of course, we'll allow them and we are still providing prime C. 6 patients are now almost 3 years, close to 3 years on the study medication from the previous study. And we are happy about that. I think that we provide them hope, maybe more than hope. So just to conclude this clinical study, we saw a reduction in the functional and respiratory deterioration, also significant changes in the ALS related biomarkers, and the study drug was well tolerated, no severe adverse events related to the drug. Now I'll discuss briefly the biomarkers. So what we did with the Mass General Hospital is an experiment to first identify the most important, the relevant biomarkers that we want and can measure. And then we tested blood samples, plasma from patients who participated in our clinical study in the phase 2A to look how that has changed over time. And the third stage of this experiment was to look at matched patients for Mass General, untreated patients, and compare them to patients that received prime C. So we're looking at this panel of biomarkers, neuroinflammation, iron accumulation, microRNAs, TDP43, and so on and so forth. We saw changes when we compared naive, meaning untreated patients or treated only with riluzole, compared to control, to healthy people. In all these markers, we saw significant changes. So now we move to the second stage and looked at our patients. But I'll jump to the third stage when we compared our patients in 2 most important biomarkers that are being used also now as a primary endpoint of the phase 2B of the paradigm study to untreated patients. So what we saw that patients that were not treated with prime C, but only with the standard of care, the levels of TDP-43 didn't change over time, over a period of 12 months, same as neuroinflammation. While when patients in our study were on prime C, in a period of 12 months, you see a very nice statistical significant reduction in TDP-43 and also in neuroinflammation. So we are very encouraged by that. Clearly, it indicates a biological activity of prime C. And now the question is, with that biological activity, what is the clinical benefit that we can provide the patients? And this question hopefully will be answered in June this year, when we will look at the results from the paradigm study. June, not this year, June 2023, in less than a year from now. So I'll describe the paradigm study, the phase 2B clinical study that we're running now. It's a study that with 69 participants, 46 participants on prime C, 23 participants on placebo. Of course, double-blind in a double-blind manner for 6 months. And after this period of 6 months, we will enable and provide prime C to all the patients. All the participants will receive prime C. As long as they wish to receive prime C, we will supply it. And this study is being conducted in Israel. It started already and enrolls patients in Israel. Soon enough, it will be also conducted in Italy and in the United States. The first and main site in the United States is planned to be Mass General Hospital, but it is likely that we'll open another clinical center in the US. And we are doing efforts, because I'm sure that for most of you this study is not relevant, we are doing efforts to start an EAP program as soon as we can. Of course, we have limited resources, but we will do whatever we can to start this program. And whenever we have enough resources, to add more and more participants who would love to, who would want to join the EAP. Of course, expanded access program, which I'm sure you're all familiar with the EAP. Just to mention about the paradigm study, what we are looking at. So I mentioned that the primary endpoints are the two biomarkers that we mentioned before, that we already obtained in the small clinical study, statistical significant results, which is the neuroinflammation and TDP-43. We want to see, again, reduction and statistically significant reduction in these markers compared to patients on placebo. The secondary efficacy endpoints are, of course, ALS-FRS, slow vital capacity, and other questionnaires. And we're looking, of course, safety and tolerability as well. As mentioned, the study is being conducted in Israel, Italy, and the US. And we call it paradigm because we believe that it's a shift in the paradigm when we are targeting multiple mechanisms in this complex disease. The Rubik cube represents the complexity of the disease, and we believe that now we shift the paradigm when we tackle multiple mechanisms, and hopefully it will be shown in the results. Just to mention where we are heading from here, so we are also moving forward the preclinical studies for Alzheimer's and Parkinson's, but again, the most important program for us is ALS, and that's the lead program as well with prime C. We expect to have top-line results from the phase 2B study by May or June 2020. To complete enrollment by the end of this year, complete enrollment of the participants, and by May or June 2023 to have top-line results from the phase 2B. To date, what we obtained is the, of course, initiation of the study. We started it on time as we planned. PK study, now we are running a multi-dose study to enable the opening a site in the US, in-life 90-day tox study to present the data to the FDA and enable a phase 3 clinical trial afterwards. And the third stage of the ALS biomarkers, which is super encouraging we got these positive results with presenting that we have clear difference between participants on Prime C as opposed to untreated patients. So with that, I will conclude for now, and of course, I welcome any question that you may have. Thank you for listening.
Well, thank you for the presentation. I feel like that was actually very informative, and we've got quite a few questions. So myself and Casey will be asking them to you and having you answer them. And we're going to kick off with probably the one that's most popular. How can PALs join your studies in the future?
Sorry, how can? Sorry?
Oh, sorry. How can people living with ALS join and partake in your studies?
So as you all know, in a clinical study, there are, of course, inclusion-exclusion criteria. People who are eligible for the study and live in the area of the clinical centers for the paradigm study, of course, we welcome them, their participants in the study. Other people who are not eligible or are not in this area, we will do all the efforts to start an EAP program. Of course, at first it will be very limited. We hope that at a certain point we will apply to the ALS Act and trying to receive a budget so we can extend and expand these programs for people who are not eligible for a clinical trial, so they can receive Prime C. Of course, we will not supply any placebo in these programs since it's an EAP. We'll do all the efforts to give it to people who would like to receive this. We are exploring this. We are trying to do it as soon as we can.
Hi, Alon. Thank you for your presentation. My first question is, what impact, if any, have you had on IL-17 and GM-CSF levels?
I'm sorry. Sorry, can you please repeat?
Yeah, what impact, if any, have you had on IL-17 and GM-CSF levels?
The question is, what impact has there been, if any, on IL-17 and GM-CSF levels?
I don't think that we have the answer. I don't think that we looked into these specific markers.
Talking about Prime C, is it a possible therapy for pre-symptomatic patients with C9orf72 mutation?
It's a great question. We don't know. Of course, once we will show efficacy, we will do another study for pre-symptomatic people with mutants, with known mutants. But we need first to show that we have the required effect on people with symptoms already.
That's good to know. It's going to be on the future of the mind site there. I'm going to just jump in a little bit. Casey's having some mic difficulties. We'll get that worked out. But moving right along, how close do you view, from what you've seen with your studies, are we as a community close to finding an approved cure or even just a treatment?
Wow, that's a tough one. I will refer first to NeuroSense and then to the field itself. I think that there is a huge potential here. We cannot promise anything, of course, but we see this biological effect that we have, and we believe that with the improved formulation and optimized doses that we're using now, we may view something that can actually slow down drastically the disease for patients. We do think, however, that not all the patients will gain the same effect. So some patients might not benefit from the drug at all, maybe. Other patients might benefit a lot from the drug. What we are doing now is also trying to identify who are the patients who will be responders super responders and who are the patients who will be non-responders, so they will not waste their time with a therapy that won't assist them. I think that basically in the field, there are lots of companies, many companies, some of them look quite promising. I think part of them also participated in the Healy trial, in the Healy platform trial. Soon enough, they will reveal the results. So hopefully they will show something in addition, of course, to Amylyx that can provide a good effect, good benefit for the patients, and it might become another treatment option.
A lot of possibilities. Specifically the Prime C, I know the trial was about a 12-month.
James, I'm sorry, I just want to add something.
Sure.
Since you said lots of possibilities, thanks to you guys. It's thanks to you, because when the Ice Bucket Challenge started, there was a blossom of lots of researchers and companies that popped out suddenly, because there was more information due to the research that started in universities. I think that to some extent NeuroSense is also a cause of this phenomena of the Ice Bucket Challenge. So thanks to people who are active in the field, who care enough, who are pushing the field, and thanks to organizations like Everything ALS, there are more and more options now, and more hope that some of the activity that is going on now will translate into a treatment option.
I know we're all looking for another Ice Bucket Challenge, because the hope that we've seen come from it has been incredible. Speaking of hope, after the 12 months of the trial period with Prime C, have you seen the results plateauing or continue to improve? Have the rates of decline remained better than untreated patients?
Okay, that's a very tough question for me to answer, because I'm sharing information that is not based on pure data. And why? Because after 12 months, since the 12 months were completed and till now, of course. So the study now that is being, we are just supplying the drug now. Basically, it's an investigator-initiated study that continued the phase 2A study. So it's all basically what I hear from the PI that runs the study, about 6 patients that are still taking the study medication after almost 3 years. And basically what I know about them is not data, not the ALS-FRS now as opposed to what was there a year ago and 2 years ago, but that they are very stable and they look good, and they feel good about it. They come to the clinic every 3 months to receive more of the drug. And that's all I know basically from what I've been heard. But the data is not ours.
Well, I think the fact that they choose to stay on the drug for 3 years is telling. The improvements from Prime C, I know you've only got the data, but has it been observed in both sporadic and familial ALS cases, or just one over the other?
So all we know is just one person with a mutant. All the other were sporadic patients. So it's hard to tell according to just one. So basically we are referring to the sporadic patients when we are talking about these changes.
Good to know. Have you studied any formulation which can be used with a feeding tube? I know you talked about it being a time release format as of right now with the Prime C.
So not yet. It's very complex and costly to achieve the same characteristics of the current formulation for a formulation that can go into a feeding tube. Once we'll see that we do provide benefit to the patients, then we'll start working immediately on a new formulation for people who are in more advanced stages and cannot swallow a tablet. Although I have to mention the tablet is very small, but of course, at a certain stage, people cannot swallow anymore. So we will do all the efforts to develop something that they can take via a feeding tube. But first we want to see that we have the required effect.
Switching gears here a little bit, could you explain the importance of biomarkers and maybe just give a little bit of a rundown of what they are?
Sure. So biomarkers are basically parameters, biological parameters such as enzymes and proteins that are well known with regards to a certain disease. Also with ALS, there are several biomarkers, several proteins or enzymes that are well characterized, and we know that either they dysfunction or they accumulate, or each one and its own story. We know, for example, that TDP-43 is something that is common to many, if not all, ALS patients. It accumulates in motor neurons, and eventually it causes it's a part of the cause of the degeneration of the motor neurons. And so what we are looking at when we're talking about biomarkers is to see that if a certain protein is toxic and it accumulates in a cell, we want to see that we can reduce it. That's what we're looking at. Of course, it's not TDP-43, maybe the most important one, but it's not the only one. We were looking at prostaglandins, which reflects neuroinflammation. We want to see reduction in neuroinflammation as well, of course, and other biomarkers that are part of this complexity of ALS.
That was, I think, a very easy way to understand it. Going off the same topic of biomarkers, could you maybe explain what the difference is between digital biomarkers and the non-digital forms?
Sure. So I think that maybe it's more right to say, and Indu, tell me if you agree with me, digital markers, not digital biomarkers, but digital markers. So it will be markers that comes from a digital platform. So something that can measure, for example, we started a collaboration with a company called Neuralite. They are measuring eye movements, micro movements of the eye. So participants are looking at the screen and they get a task to look at a certain point. They should follow the point with their eyes, and a software collects all the data about the micro movements of their eyes and analyze it eventually, and can tell if the patient declined or what is status. Basically, it's like providing the patient with I should say some kind of a score like the ALS-FRS, but it's more objective. It's calculated very accurately via an AI program.
Thank you.
Right,
I just wanted to add, Alan, is like what you said, the speech study we do, or GATE, like taking a physiological data and using computer science to really figure out with machine learning and AI to see the pattern so we can do the biomarker. So that's kind of what we call digital biomarkers as opposed to just biology.
Right. When we say classic biomarkers, so it's something that you measure via CSF or serum, blood plasma, or any other method, but biologically, something that you can measure biologically.
I know we just talked about TDP-43, but will Prime C also be relevant to C9orf72 as well?
We believe so. We don't know it, but we believe so. And when we say TDP-43, we don't mean that it's relevant only to people with mutant of TDP-43, but it's, again, TDP-43 is something that, all cross-board, that affects all patients. This toxic protein affects all patients, as far as we know, of course, or many of the patients, sporadic patients. and it accumulates in the motor neuron cells and degenerates.
Specifically, I believe this is our last question for Prime C. Does Prime C affect, I'm going to butcher the name of this, so forgive me, the riluzole levels in ALS patients?
We didn't measure that, but we did provide, of course, concomitant treatment. So many participants in the previous study and also in the current study can take riluzole and Prime C together. So it's not an issue. We just need the participants, the patients, to be stable on riluzole for 30 days, for 1 month. And after that, if everything is well with the patients and they feel good with riluzole, they can take Prime C.
Perfect. We're going to switch a little bit to paradigm here. And since we're talking about riluzole, what interaction with paradigm and riluzole causes the increase in benefit, in your opinion? And do you think there will be a similar beneficial reaction with Radicava?
So we don't know yet if riluzole and Prime C are better than just Prime C alone or just riluzole alone. What we'll do once we conclude the study, once we have the top line, the results from the study, we will compare. I think that most of the patients eventually will be on riluzole. That's how it is in the US, in Israel, also in Italy. Many patients are taking riluzole. So what we are going to do eventually is looking at participants who didn't take riluzole, but were on Prime C, compare them to people who were on riluzole, but also on placebo, not on Prime C, compare them to people on riluzole and Prime C, and to people on placebo without riluzole. So all the 4 groups, and to see if we have differences between the results there. And to see if we find that maybe riluzole in Prime C is better than just riluzole alone, and maybe it's better than just Prime C alone. Could be. We don't know yet.
It'll be an interesting study at one point.
It is. Although the numbers are quite small, but we hope that the magnitude is enough to show and present these results. And you mentioned Radicava or Edaravone. Same with the Edaravone, although much less patients are receiving Edaravone currently nowadays. But we'll do the same. We look at the results the same way.
No stone left unturned. Make a little easier question here for you. Can you comment on the eligibility criteria for Paradigm?
Sure. So the main ones the participants should be 30 months up to 30 months from symptom onset, up to 75 years old, with ability to swallow. So the score should be 3 in the ALS-FRS when in the question of swallowing. What else is important to know? I think that these are the main criteria that we're looking at.
I just see it in the chat right now, and it goes with this. It's okay. Is frontotemporal dementia an excluding factor or no?
Yes, it is. The participants should have only ALS, so it will be more clear when we analyze the results. I'm sorry. Each time I say such things, I'm sorry for all the people who are not eligible for the study. I'm really sorry about it. But for future patients and also current patients, but for the benefit of research, we must limit some factors so the results will be more clear to us eventually. I will mention also that the ALS-FRS should be above 25, equal or above 25, and the slow vital capacity equal or above 60.
has NeuroSense given thought to participating in the Healy platform trial? Why or why not?
Yes, we're giving thought about participating in the Healy trial. Maybe there will be some news sometime soon, but currently it's not something that that has materialized yet, but in discussions. That I can say. It's something that we are considering, we are discussing.
Then we will keep our eyes peeled for that. Would you say that entering the US market for studies is more difficult because of the FDA, or is it somehow easier?
With regards to clinical studies?
I believe so, just in general, clinical studies, or maybe even approval for drug usage like Prime C once it passes the stages of trials.
I think that once we pass the stages of the trials, the US might be, it's quite difficult to say. Of course, when we are looking at Amylyx, we see that the Canadian approved the Amylyx drug, their combination drug. The FDA didn't approve it yet. They might in September, but they dwelled on it and delayed the PDUFA date. On the other hand, when we are looking, when you are saying, when you're asking this question, of course, you compare between different territories. So the other territory to compare to is Europe, and I think that the Europeans are more strict about it, and it's more difficult to get a study medication approved. There are other boxes to check before you submit your application for approval in Europe.
That's very interesting. I, for some reason, always had it the other way around. Speaking of the FDA, if the upcoming clinical trials continue to be successful in reducing the progress of ALS, how soon do you think it could be approved by the FDA for use by ALS patients?
Currently, our estimations are talking about 2025. If we are super optimistic and super fortunate and empowering the current study, the paradigm study, of course, it can be sooner, not tomorrow in 2023, but maybe 2024. Otherwise, we on the safe side, what we're saying is 2025, not to provide the false hope.
Well, it's better than 10 years, so we'll take it. I know it's very late where you are. We want to be very respectful of your time. I want to thank you.
It's a non-issue, really. I enjoy being here. I enjoy communicating with you guys. So it's super important for me to provide as much information as I can.
And boy, did we get a lot tonight. I'm going to make this the last question, and since it just popped up on my chat here, are you still using the old criteria, or have you begun to use the Gold Coast criteria in regards to eligibility?
I'm not sure I was following. Can you explain the question?
Boy, you and I are going to be in the same boat here. The criteria in order for eligibility, do you use the Gold Coast criteria?
I'm not sure what is the Gold Coast criteria.
LS Escorial revised versus the GDC.
So we are using the Escorial, of course, to define who's a probable, who's a definite, and ALS patient. So, yeah, sorry for not understanding before.
That's okay. I didn't either. Now I learned something new. Well, we, believe it or not, went through almost 20 questions there. So I'm going to invite you to stay along if you'd like, but I know I've got a lot going on in your life with testing everything. So we're going to have this portion be the open forum. I'm going to hand it back to McFinn. I do want to thank you once again. This was an incredible presentation. I feel like it's so hopeful, showing how much positive is coming from this. So thank you so much.
Thank you, James, and thank you all the participants. Thank you for being here, for listening, and thank you for participating basically in the Everything ALS programs and being active in the field. You are our energy. So thank you very much.
Thank you, Alon. I got a couple of questions I'd just like to run by you. And it's obvious that you have a passion for what you're doing. So where did you gain this passion for ALS?
I think that that it started with Shai, with knowing Shai, such a special person who did so much for the field. And then more and more people, since once I got into the field, more and more people, very special people that are active in this field. And that is, again, that's our fuel. That's what gives us the motivation to do what we are doing day in and day out. It's a horrible disease. It's a fatal disease. The people that are fighting it, they are amazing. The people who are active and fighting this disease are amazing. And also the people, the caregivers and family members of people who are still, even after they lost their loved ones, they are still here. They are still doing what they are doing. I admire them.
Wow. Yes, we all admire them. Can you ask one? I got one more question. And can you describe how you felt like our group was listening to you tonight?
I think that according to the questions, I was of course focusing with delivering the presentation, but with these questions, so many questions and very good questions, I feel like, you know, it was a great honor for me to present and to answer the questions as much as I could, of course, answering these questions. I think that you are super attentive, guys, and I really appreciate it.
Thank you very much. You know, I have one person with their hand up. Brenda, you want to take yourself off mute? There you go.
Hello.
Hi, Brenda.
Hello, Brenda.
I have a question. I have HSP, and it's in the family of ALS. It's hereditary spastic paraplegia. Would we be able to try any of these medications?
I don't know to tell you, you know, if it's something that you'll be eligible for the paradigm study, but as I mentioned, with regards to an EAP program, we'll do all the efforts to have the criteria as broad as we can, to provide it to any patient that we believe that Prime C will, of course, not risk him if he's not sensitive to one of the compounds, he or she. I don't see any reason why to avoid providing it.
Thank you.
Thank you, Brenda. And with that, Alon, we're going to open up our open forum and see what sparked everybody this evening during this conversation that you gave us.